Technobotany by Zura. In this video, we'll explore the revolutionary gene editing technology, its principles, history, various applications, recent innovations, and future outlook. What is CRISPR-Cas? CRISPR-Cas, short for clustered regularly interspaced short palindromic repeats and CRISPR-associated proteins, is a revolutionary tool in modern genetics. Originally discovered as a natural immune system in bacteria, it helps these microorganisms defend themselves against invading viruses by recognizing and cutting their DNA. Scientists have adapted this system for gene editing, allowing precise and targeted alteration of DNA in living organisms. With CRISPR-Cas technology, researchers can add, remove, or modify genes with remarkable accuracy, opening new possibilities in medicine, agriculture, and biotechnology. History of CRISPR technology. CRISPR was first discovered in 1987 in E. coli as unusual DNA sequences. In 2007, scientists identified CRISPR as an adaptive immune system in bacteria, enabling them to defend against viruses. The breakthrough in gene editing came in 2012 when Jennifer Doudna and Emmanuel Charpentier demonstrated how CRISPR-Cas9 could be used to precisely edit genes. Their pioneering work revolutionized genetics, leading to the Nobel Prize in Chemistry in 2020. This historic development has paved the way for countless scientific advancements. Mechanism of CRISPR-Cas systems in bacteria. The CRISPR-Cas system in bacteria functions as a powerful adaptive immune mechanism, protecting the cell from invading viruses. Its action unfolds in three main stages: adaptation, expression and processing, and interference. During the adaptation phase, the bacteria capture small fragments of viral DNA, known as spacers, and integrate them into the CRISPR locus with the help of Cas proteins. These spacers serve as molecular memories of past infections. Next comes expression and processing. The CRISPR array is transcribed into a long pre-cRNA, which is then processed into shorter cRNAs by Cas proteins. Finally, in the interference stage, the cRNA-Cas complex patrols the cell, searching for foreign DNA sequences that match the stored spacers. When a match is found next to a PAM sequence, the Cas enzyme binds and cleaves the target DNA, effectively neutralizing the invader and protecting the bacterial cell. Naturally found in bacteria, CRISPR-Cas has been adapted into a powerful genome editing tool. Let's look at the main components that make this system work. First, we have the guide RNA, which acts like a GPS. It finds and binds the matching DNA sequence in the genome. The Cas nuclease. This is often compared to a pair of molecular scissors. It cuts DNA at a precise spot, guided by the guide RNA. But Cas can't cut just anywhere. It needs a short signal sequence right next to the target, called the PAM motif, or protospacer adjacent motif. Each type of Cas enzyme recognizes its own PAM pattern. Guide RNA, sgRNA, and its structure. The guide RNA is what directs the Cas nuclease to the right spot in the genome. It's a short stretch of RNA that base pairs with the matching DNA sequence to bring the nuclease exactly where it needs to cut. The sgRNA merges two natural components: a 20-nucleotide cRNA sequence that determines the DNA target, fused via a hairpin loop to the tracer RNA scaffold that enables Cas9 binding. The sgRNA Cas9 complex scans genomic DNA for a complementary sequence followed by a PAM motif, typically NGG, for recognition. Once these conditions are satisfied, Cas9 catalyzes a precise double-strand break, typically 3 bases upstream of the PAM motif. Principles of sgRNA design. Designing an sgRNA involves several important principles. First, the target site should be within the coding region, preferably early in the gene, for effective gene knockout. Avoid SNPs, repetitive regions. Next, the sequence should lie just upstream of a PAM motif. GC content should ideally be between 40 to 60% for stable binding. And most importantly, we must minimize off-target effects by checking for similar sequences elsewhere in the genome. Check specificity. Now, we need to assess how specific our candidate sgRNAs are. There are several tools available, such as CRISPR, ChopChop, and Benchling, that allow us to input our sgRNA sequence and check for similar sequences across the genome. Evaluate efficiency. After filtering for specificity, we evaluate the efficiency of the remaining guides. Tools often use scoring models like the Dunch 2016 score to estimate how well each sgRNA will guide Cas9. High-efficiency guides increase the chance of successful editing. In some cases, data like chromatin accessibility can also inform our choice. Different types of Cas proteins in genome editing. The functional architecture of Cas9. The next component is Cas protein. Here we see the Cas9 nuclease, a vital enzyme in the CRISPR gene editing system. The enzyme consists of two main parts, the recognition lobe, which binds the DNA, and the nuclease lobe, which contains two catalytic domains, the HNH and RUVC domains, that cut the DNA strands. The HNH domain is responsible for cleaving the DNA strand that is complementary to the guide RNA. The RUVC domain, in contrast, cuts the non-complementary DNA strand. Together, the HNH and RUVC domains ensure precise and coordinated cleavage of both DNA strands, creating the characteristic blunt-ended double-strand break that underpins CRISPR genome editing. The REC1 domain plays a major role in recognizing and stabilizing the RNA-DNA hybrid, the R loop, that forms during target engagement. It acts as a molecular scaffold, sensing conformational changes that occur when the correct target is recognized. Target recognition also depends on the PAM interacting domain, or CTD. The PI domain ensures that Cas9 only binds DNA sequences adjacent to a specific PAM motif. Bridging the recognition and nuclease lobes is a flexible structural element known as the bridge helix. The bridge helix communicates activation signals. When Cas9 binds the correct RNA-DNA hybrid, the bridge helix helps transmit the signal that activates the HNH and RUVC catalytic domains, ensuring that cleavage occurs only upon precise target recognition. Cas12a. Cas12a, also known as CPF1, is a powerful genome editing enzyme that recognizes a TTTV PAM sequence, where T stands for thymine and V can be adenosine, cytosine, or guanine. Unlike Cas9, Cas12a makes staggered, or sticky-end cuts in DNA, which allows for more precise and seamless gene insertions or modifications. Another advantage of Cas12a is that it requires only a short crRNA, eliminating the need for a tracer RNA, simplifying the editing system. Because of its flexibility and precision, Cas12a is becoming increasingly popular, particularly in plant genome editing, where fine control over DNA modification is essential. Cas13. Cas13 is a unique member of the CRISPR family that targets RNA molecules instead of DNA. It recognizes specific RNA sequences through a guide RNA and then cuts them with high precision. Unlike other CRISPR systems, Cas13 does not require a PAM sequence, giving it greater flexibility in target selection. Some Cas13 enzymes prefer targets with a given single-base protospacer flanking site, PFS, sequence. Once activated, Cas13 can also exhibit collateral cleavage activity, cutting nearby RNA molecules, a feature that has been creatively used in diagnostic tools like Sherlock for detecting viruses and other pathogens. The HEPN-activated conformation of Cas13 is a general nuclease, which is capable of cleaving the RNA molecule. Because of its RNA targeting ability and versatility, Cas13 is becoming an important tool for gene regulation, antiviral therapies, and molecular diagnostics. Cas14. Cas14 is one of the smallest and most compact CRISPR enzymes discovered so far. Unlike many other CRISPR systems, Cas14 is capable of targeting and cutting single-stranded DNA, ssDNA, without requiring a PAM sequence, making it exceptionally versatile for precise genetic manipulation. Its small size allows for easier delivery into cells and viral vectors, which is a major advantage for gene therapy and genome editing applications. As research advances, Cas14 is emerging as a next-generation CRISPR system, combining efficiency, flexibility, and compact design for future biotechnological innovations. Engineered variants of Cas9. Over time, scientists have developed several engineered variants of Cas9 to enhance its performance and adaptability in genome editing. One notable variant is SA-Cas9. It is smaller in size compared to the standard SP-Cas9, making it especially suitable for delivery through compact systems such as viral vectors, where space is limited. Other engineered versions, including ESP-Cas9 and SP-Cas9HF1, were designed to reduce off-target effects, improving the precision and safety of CRISPR-based editing. dCas9, or dead Cas9, contains mutations in both the RUVC and HNH domains, allowing it to bind DNA without cutting it, a feature used in CRISPR interference and activation, or CRISPR-I and CRISPR-A. Meanwhile, nCas9, known as the nickase Cas9, has a mutation in just one nuclease domain. It cuts only a single strand of DNA, enabling more precise editing and reduced off-target effects. PAM motif, the entry code. The PAM motif is a short sequence in the DNA that's absolutely required for CRISPR to work. The Cas nuclease can only bind and cut if this sequence is present right next to the target site. Different nucleases recognize different PAM sequences. For example, SP-Cas9 requires an NGG sequence, while many Cas12a variants look for TTTV. Editing strategies. Once the system is in place, editing can happen in different ways. The traditional method is a DNA cut, followed by the cell's repair processes, which can add or remove bases, or incorporate a donor DNA template for precise edits. More advanced versions include base editors, which change one DNA letter to another without cutting, and prime editors, which can write in new DNA sequences more flexibly. How does CRISPR-Cas work? The CRISPR-Cas9 system begins with designing a guide RNA, gRNA, that matches the target DNA sequence. The gRNA forms a complex with the Cas9 protein, creating a molecular scissors. This complex scans the genome to recognize and bind to the specific target sequence. Once Cas9 binds the DNA at the target site, it induces a double-strand break, about 3 to 4 nucleotides upstream of the PAM motif. The cell's natural repair mechanisms respond, either repairing the break with small mutations or through precise editing when supplied with a repair template. This process enables targeted genetic modifications with high efficiency. Delivery vehicle. Of course, to actually make this work in cells, we need a delivery vehicle. This could be a plasmid, a viral vector, or a protein RNA complex. Supporting elements, like promoters, nuclear localization signals, and selection markers are added to make sure the system works efficiently once inside the cell. Readout and validation layer. To quickly confirm if CRISPR editing has occurred, scientists often use PCR combined with gel electrophoresis, or the T7E1 assay to detect small changes in DNA. For precise identification of the exact modification, Sanger sequencing or next-generation sequencing provides detailed, base-by-base information about the edit. When it comes to quantifying how efficiently the edit was made across a population of cells, techniques like quantitative PCR or digital PCR are employed. Finally, for functional validation, researchers use phenotypic analyses or protein-based assays, such as Western blotting or reporter assays, to confirm that the genetic change leads to the desired biological outcome. Applications in medicine. CRISPR offers transformative potential in medicine, including the possibility to cure genetic disorders like sickle cell anemia and cystic fibrosis. It is advancing cancer research by enabling precise genetic modifications. CRISPR also shows promise in antiviral therapies for infections such as HSV and HIV. Furthermore, it paves the way for personalized medicine, tailoring treatments to individual genetic profiles. Applications in agriculture. CRISPR is revolutionizing agriculture by developing drought and pest-resistant crops, ensuring food security. It also helps improve crop yields and enhance nutritional content, addressing global nutritional needs. Additionally, CRISPR enables the creation of disease-resistant livestock, promoting healthier animals and more sustainable farming practices. These innovations are shaping a more resilient and efficient agricultural future. Applications in research. CRISPR is transforming research with its versatile applications. It enables functional genomics studies, helping scientists understand gene roles and interactions. In disease modeling, CRISPR allows precise creation of models to study various conditions. It also facilitates the development of transgenic organisms, advancing agriculture and medicine. Additionally, CRISPR accelerates drug target discovery, paving the way for new treatments. Recent innovations. In the rapidly evolving landscape of genetic engineering, CRISPR technology continues to push boundaries with groundbreaking innovations. Among these are base editing and prime editing, which have revolutionized the precision with which genetic modifications can be made, allowing for accurate corrections of mutations without introducing double-strand breaks. Another significant advancement is in CRISPR-based diagnostics, such as Sherlock and Detector, which utilize the specificity of CRISPR systems to detect pathogens and genetic variations swiftly and accurately, transforming disease detection and biosensing. CRISPR is also making strides in epigenetic modifications, enabling scientists to alter gene expression without changing the underlying DNA sequence. This opens new frontiers in treating complex diseases and understanding gene regulation. Lastly, ongoing delivery improvements are enhancing the efficiency and safety of CRISPR systems, ensuring that these powerful tools can be effectively and precisely delivered to target cells within living organisms. Ethical and safety concerns. While powerful, CRISPR raises important ethical and safety concerns. Off-target effects, where unintended edits occur, pose risks to safety. Germline modifications, which can be passed to future generations, spark ethical debates about morality and human integrity. Human embryo editing especially prompts discussions on consent and potential misuse. Finally, biosafety regulations are vital to ensure responsible development and prevent misuse. Balancing innovation with moral responsibility is essential to use CRISPR ethically and safely. Future perspectives. Looking ahead, CRISPR holds great promise for advancing clinical trials and developing new therapies. As gene editing tools continue to expand and improve, their potential to treat and possibly cure genetic diseases grows. The establishment of comprehensive global regulatory frameworks will be crucial to ensure safe and ethical use of these technologies worldwide. Ultimately, these advancements offer the exciting possibility of eradicating many genetic disorders, transforming healthcare for future generations. I hope you enjoyed this video.